Okay, chapter 4 is genetics and cellular function, so let's talk about that. The first thing we need to understand is that everything that we're going to talk about in here is basically related to the nucleus. That is why the main topic in here is the nucleic acids. As you can see here, Na stands for nucleic acid, and here Na, nucleic acid. So there are two main types of nucleic acids, RNA and DNA. Where are they located? They are located in the nucleus. Okay, well, that's it. So if we scroll down, in here is just history, as you can see here, 1844, 1900. So this is just history regarding the DNA and the RNA. You don't need to learn basically anything about it regarding history. But we can go directly into the DNA structure and function. The DNA structure is composed of a nucleotide. As you can see down here, nucleotides are the building blocks of DNA as well as RNA. DNA, the main job of the DNA is to transfer the genetic information from one generation to another. So basically, you are here because your dad and your mom gave you the DNA, which is later on, the DNA is going to provide the genetic information that is going to make you as a person. So we said that the DNA is composed of nucleotides as well as RNA. So all the nucleic acids are made of nucleotides. The nucleotides are composed of three main areas, three main things. They're composed of sugar, a phosphate, right here, and a nitrogenous base. So what are the components or what are the parts of a nucleotide? Sugar, phosphate, and a nitrogenous base, or a base that contains nitrogen. So which ones are the bases that contain nitrogen? Cytosine, thymine, adenine, guanine, and you're going to see later on that we have another one which is uracil. So if we scroll down here, so in here you have the graphical representation of one nucleotide where you have a phosphate, you have a sugar, in this case deoxyribose, and you have the base that contains nitrogen, that is going to be adenine in this case. So we have three different parts. Okay, as you can see here, you can see the different individual nitrogenous bases. As you can see here, there are 5: adenine, guanine, cytosine, thymine, and uracil. Uracil is used in RNA, and we're going to see that in a little bit. So we keep scrolling down. So the DNA is this one right here. So you see here, DNA has two strands, one strand, two strands, or two chains. So the two chains are going to be the main characteristics of the DNA. RNA has only one strand, so one strand right here. So where do you get the DNA? Basically, the DNA is going to contain the genetic information that your dad and your mom gave you, and this is going to have two strands. RNA can be created or will be created from the DNA. Again, the main difference is that this has two strands, RNA has only one. Another difference is that RNA contains uracil, the letter U, but DNA does not. As you can see here, the combination does not have U. We're going to talk about it in a little bit. So we'll keep scrolling down right here, and it says in here that the combination of the nitrogenous bases, the base pairs, is going to be always A with T and C with G in the case of DNA. So A with T and C with G, always in the case of DNA. So let's keep scrolling down. So this is history again of the DNA. More history. You don't need to know any of that. That's basically it. History. So then we continue and we have DNA, as we said, that is the way in which we transfer information in the from one generation to another using genes. And the genes eventually are going to give us the proteins that are necessary to create a human being. So how do you see this structure? How do you see this DNA? As we were saying, DNA has a double helix. As you can see here, one strand, two strands that are going to start twisting, and it's going to form this structure called a double helix. When you put together this double helix, you are going to get something called a chromosome. So if you scroll down here, that's why one of the first things that we're going to have right here is chromosome. Okay, another very important concept that you need to know is chromatin. When the cell is working, when the cell is developing, the cell is doing whatever the cell needs to do, then you're going to see chromosomes. I'm sorry, you're going to see chromatin. And when the cell is dividing, you're going to see chromosomes. So there are two main states, let's say, that your cell is going to have. One state is what the cell does every single day. And another state is when the cell decides that it needs to divide. When the cell is working every day, doing or fulfilling the functions that it needs to perform, then you are going to have chromatin. Eventually, that cell needs to divide and create another cell. So during the process of cell division, you're going to see chromosomes. Later on, we're going to see this with a little bit more detail. So if we scroll down right here, then you can see the two different structures, the chromatin and the chromosomes. Again, we said that there are basically two stages that the cell is going to go through during its life. One state is when the cell is doing the normal functions that every day needs to perform, and the other one is that when the cell needs to divide. So when the cell is performing the jobs that it needs to perform every single day, what you're going to see, the genes that you're going to see that your dad and your mom gave you, they're going to be in this form right here, which is the chromatin. You see it has no particular shape. It's just like spaghetti. You see, it's all of them in here, all around, nothing in particular you can say about it. But then when the cell is going to divide, all this chromatin is going to condense. It condenses, and eventually it's going to give you this structure right here at the bottom. After the condensation, it's going to give you this, which is going to be the chromosome. The chromosome in here, as you can see, it has an X shape. Each arm, so for example, this piece right here, that's one, and this is the other one. Each one of these arms is a chromatid. So don't confuse chromatin, chromosome, and chromatid. Chromatin is when it's all dispersed, no condensed. And this happens when the cell is when the cell is performing its normal jobs every single day. Chromosomes are when this chromatin gets condensed, and you can see that when the cell is about to divide. Now, once you have this chromosome, each arm of the chromosome is called a chromatid. So I hope that that is clear. So if we keep scrolling down, you can see here it says compactation, because the chromatin needs to get compacted in order to get a chromosome right here. So if we keep scrolling down, then you see the DNA, and we have in here sister chromatids. As we said, the chromatids are the arm of the chromosome. The chromosome is going to have two arms. One arm, each arm is a chromatid. So as you keep scrolling down, you can see here again the characteristic of a chromosome, which is a chromatin that has been condensed, and each arm is a chromatid. The middle is called the centromere, this part right here in the middle. Okay, let's keep scrolling down. So right here they are telling you a comparison between DNA and RNA. Main difference: sugar. The DNA has deoxyribose, RNA has ribose. That's why RNA, the R represents the ribose. Ribose, nucleic acid. This one is deoxyribose, this is deoxyribose, that's the D, nucleic acid. Okay, after that, difference, another one is the bases. We said always A with T and C with G in the case of DNA. In the case of RNA, it's always A with U and C with G. So in the case of RNA, you have U, you never have T in the case of RNA. You always have U instead of T. So the combination of, in the case of RNA, is going to be A with U and C with G. You can see the other different characteristics that you have in here, but the ones that I would like you to understand for this class are these two, and also this one, which is how many arms you have, or how many pieces you have. So this one is always 2, as I showed you before, and this one is only 1 strand. 2 strands that are going to twist. This is just 1 strand. The RNA comes or is created from the DNA. If we scroll down from the table, we're going to see here the RNA structure and function. We said that RNA, it has a single strand, and it has uracil, the letter U, instead of letter T. And also RNA has ribose instead of deoxyribose. And where the RNA comes from? From the DNA. What is the job of the RNA? Basically to take the information from the RNA, to take the information from the DNA and create proteins. So where the RNA comes from the DNA. What's the job of the RNA? Is to take the information from the DNA and create proteins. How many different kinds of RNA we have? We have three. We have RNA messenger, RNA ribosomal, and RNA transfer, as you can see right here. This is RNA transfer, tRNA, rRNA for ribosomal RNA, and mRNA for RNA messenger. They all three work together in order to create proteins. That's the main thing about it. If we keep scrolling down, as you can see in here, it says uracil, as I mentioned. That is very important. So then, as I was telling you, the tRNA, RNA, and mRNA are going to work together, and we're in order to create proteins. There's a process in which the proteins are created, and that's something that we're going to cover later on in this chapter. Again, what's the job of the RNA? Basically, to get information from the DNA to create, as it says right here, to create proteins, to synthesize proteins. The next item that we have in this chapter is genes and their action. So what is a gene? So basically a gene is a piece of your DNA that using RNA is going to codify, is going to build proteins. So if we scroll down right here, it says what is a gene, and in here it's going to talk about history. You don't need to know that part regarding history. So if you scroll down, so after the history, it says in here, we will define gene as the information containing segment of DNA that codes for the production of molecule of RNA in which eventually you are going to create proteins. Okay, so that's basically what I said. So how many genes you have? Well, when you have the genetic information that your dad and your mom are going to give you, what you're going to get is chromatin, we said. When you put together all this chromatin that your dad and your mom gave you, you're going to get chromosomes. How many chromosomes you're going to get? You're going to get 2 sets of 23 chromosomes or 46. Okay, that's what you're going to get. 23 pairs or 46 chromosomes. So all the information that you get in these 46 or 23 pairs of chromosomes is your genome. That's what you got from your dad and your mom. If you have big eyes, it's because that's in your genome. If you have big toes, it's because that's in your genome. If you have you produce too much acid in your stomach, then that's because it's in your genome. Everything that you are is in your 46 chromosomes, and that determines your genome. Your genome obviously is different than somebody else's genome because we all are humans, but we all get different kinds of information from our parents. So let's scroll down and let's see what else we have. And it says the genetic code. The genetic code is the way you read the genome. For example, it's like the system that is used in order to read the genome that you receive from your parents. For example, a set of 3 bases, for example, UAG, that is a codon. So, for example, everybody knows that set of 3 letters is a codon. Some codons have genes, for example, to codify for insulin, and some other 3 codons, some other 3 letters, another codon, may codify for actually nothing. So again, how do you interpret all this information that you have in your genes? You do it using the genetic code. Okay, that's very important for you to understand. So if you keep scrolling down, right here you see, these are the sets of bases, U, A, C, and G. A set of 3 of them, in any sequence that you may have, depending on your genome, that's going to be a codon. Okay, in some cases you can also have it as base triplet, because triplet is what? 3. So you need to understand that later on. We're going to cover what an anticodon is. So if you scroll down right here, you see it says in here other 3, UAG, for example, may codify for something such as stop. Another set of 3 may codify for start. So, and again, all this is given to you by the genetic code. If we keep scrolling down, we have right here a table showing you examples of the genetic code. The next part that we have is protein synthesis. In the protein synthesis, there are 3 main steps. Okay, so if we scroll down, you can see here transcription. The next one is translation. Okay, very important.